Welcome to this session on antigen presentation and the major histocompatibility complex, or MHC. This topic is fundamental to understanding how our immune system recognizes foreign substances and responds to infections and transplants. The following are the learning outcomes for this session. Let's begin with a clinical case that illustrates the immune system in action. Sarah, a 28-year-old woman, presents with sore throat, fever, and fatigue that have persisted for several days. These are common symptoms suggesting infection. On examination, her physician notices swollen lymph nodes in her neck and white patches on her tonsils. The swollen lymph nodes indicate active immune responses occurring in these secondary lymphoid organs. Suspecting bacterial infection, the physician takes a throat swab for culture and prescribes antibiotics. But what's happening immunologically in Sarah's body? When bacteria infect her throat, they're captured by antigen-presenting cells in the local tissues and lymph nodes. These APCs process the bacterial antigens and present them to T cells, initiating adaptive immune responses. The swollen lymph nodes reflect the proliferation of lymphocytes responding to the infection. This case illustrates the critical role of antigen presentation in detecting and responding to infections. Understanding this process is fundamental to immunology and clinical medicine. APCs have two primary roles. First, they capture and process antigens for presentation to T cells. Antigens are foreign molecules, usually proteins from pathogens like bacteria, viruses, or parasites. APCs break down these complex proteins into smaller peptide fragments suitable for T cell recognition. Second, APCs produce signals required for lymphocyte proliferation and differentiation. Presenting antigen alone isn't sufficient to activate T cells. APCs also provide costimulatory signals and cytokines that determine whether T cells become activated and what type of response develops. There are three main types of professional APCs. Macrophages are phagocytic cells that engulf pathogens and present their antigens. They're abundant in tissues throughout the body. Dendritic cells are the most potent APCs, specialized for capturing antigens and migrating to lymph nodes to activate naive T cells. B cells can present antigens to helper T cells, particularly antigens they've captured through their surface antibodies. Understanding APCs is essential because they're the gatekeepers determining which antigens trigger adaptive immune responses. Let's examine how APCs process exogenous antigens, which means they are from outside the cell. Exogenous antigens are those inhaled, ingested, or injected from outside the body. Phagocytic cells like dendritic cells and macrophages are particularly important for processing these antigens. The process follows several steps. First, APCs engulf the antigen through phagocytosis or endocytosis, internalizing it into vesicles called phagosomes or endosomes. Second, the antigen is degraded into short peptides within these vesicles. Proteolytic enzymes break down the protein into fragments. Third, these peptides are displayed on the cell surface along with class II MHC molecules. The MHC class II molecules bind the peptide fragments and transport them to the cell surface for display. Finally, these peptide MHC class II complexes are ready to be recognized by CD4 cells or helper T cells or TH cells. The CD4 molecule on helper T cells specifically binds to MHC class II molecules, ensuring these T cells interact with the right type of MHC. This pathway enables the immune system to detect extracellular pathogens and coordinate appropriate immune responses. Now let's examine the endogenous antigen pathway, which detects intracellular pathogens like viruses. This pathway operates in all nucleated cells, not just professional APCs. Endogenous antigens are generated within cells. The most important example is viral proteins, produced when viruses infect and replicate inside cells. Tumor antigens and some intracellular bacterial proteins in infected cells also follow this pathway. Endogenous proteins are processed and presented to MHC class I molecules. These complexes are recognized by CD8 cells, or cytotoxic T cells, or TC cells. The CD8 molecule binds specifically to MHC class I molecules. The major histocompatibility complex was discovered through transplantation studies when scientists noticed that tissue grafts between individuals were often rejected. MHC gene products were identified as responsible for this graft rejection. MHC refers to proteins coded by a cluster of genes on chromosome 6 in humans. In humans, MHC is also called human leukocyte antigen or HLA because these proteins were first discovered on white blood cells. MHC molecules are displayed on cell surfaces and serve two critical functions. First, they're responsible for lymphocyte recognition. T cells recognize antigens only when presented by MHC molecules. T cells cannot recognize free antigens. They must see peptides bound to MHC. Second, MHC molecules enable antigen presentation, displaying peptide fragments from proteins for T cell surveillance. The diagram shows MHC molecules marked P, Q, R, displaying different peptides. Each person's MHC molecules are unique, like an immunological fingerprint. This explains why transplantation between individuals typically results in rejection, in which the recipient's immune system recognizes the donor's MHC as foreign. First, MHC plays a critical role in transplantation. Differences in MHC between donor and recipient are the primary cause of graft rejection. The more closely matched the MHC, the better the transplant outcome. Second, MHC is essential for immune responses, including antibody formation. Helper T cells, which are necessary for B cells to produce antibodies, must recognize antigens presented by MHC class II molecules. Third, MHC genes are highly polymorphic. This means there are many different versions or alleles of each MHC gene in the population. This extraordinary diversity ensures that populations can respond to many different pathogens, providing evolutionary advantage. Fourth, MHC molecules bind peptides and present them for recognition by T cells. This peptide binding function is what makes MHC molecules the antigen-presenting molecules of the immune system. The MHC gene complex is located on the short arm of chromosome 6 and spans approximately 3.6 million base pairs, making it one of the most gene-dense regions in the human genome. Class I genes, shown in light green, encode 3 types of Class I proteins: HLA-A, HLA-B, and HLA-C. These are expressed on all nucleated cells and present endogenous antigens to CD8-positive T cells. Class II genes, shown in dark green, encode Class II MHC proteins, HLA-DP, HLA-DQ, and HLA-drive. These are expressed only on professional antigen-presenting cells and present exogenous antigens to CD4-positive T cells. The extraordinary polymorphism is evident in the numbers. Approximately 60 HLA-A alleles, 100 HLA-B alleles, and 40 HLA-C alleles exist in human populations. When you consider that each person inherits one set from each parent, creating different combinations, roughly 5 × 10 to the power of 10 allelic combinations are possible. This means virtually every person has a unique MHC profile. This diversity is beneficial for populations, ensuring some individuals can respond to new pathogens. However, it makes finding matched donors for transplantation extremely difficult. Let's examine MHC class I structure, which consists of 2 chains. The molecule has 4 distinct regions. First, the peptide binding region forms a groove created by the alpha-1 and alpha-2 domains of the alpha chain. This groove accommodates peptides typically 8 to 10 amino acids long. The sides of the groove are formed by alpha helices, while the floor consists of beta sheets. This structure holds the peptide firmly for presentation to T cells. Second, the immunoglobulin-like region consists of the highly conserved alpha-3 domain. This domain resembles immunoglobulin structure and serves as the site where CD8 on T cells binds. This ensures that CD8-positive T cells specifically interact with MHC class I molecules. Third, the transmembrane region consists of a stretch of hydrophobic amino acids spanning the cell membrane, anchoring the molecule to the cell surface. Fourth, the cytoplasmic region extends into the cell and contains sites for phosphorylation and binding to cytoskeletal elements, helping to stabilize and position the molecule. Additionally, beta-2 microglobulin, a small protein, associates non-covalently with the alpha chain and is essential for proper folding and stability of MHC class I molecules. This diagram illustrates the complete pathway of exogenous antigen processing and presentation to helper T cells. Let's trace each step. An extracellular protein antigen, such as bacterial proteins, is first captured by the antigen-presenting cell through phagocytosis or receptor-mediated endocytosis. The antigen enters the cell within a vesicle. This vesicle, called a phagosome or endosome, fuses with lysosomes containing proteolytic enzymes. These enzymes degrade the protein into peptide fragments of varying lengths. Meanwhile, MHC class II molecules are synthesized in the endoplasmic reticulum. A special protein called invariant chain occupies the peptide binding groove, preventing it from binding peptides prematurely. The MHC class II molecules are transported in vesicles that fuse with the vesicles containing degraded antigen peptides. The invariant chain is removed, allowing antigenic peptides to bind to the MHC class II groove. The peptide MHC class II complex is then transported to the cell surface where it's displayed. Helper T cells with appropriate T cell receptors recognize this complex. The CD4 molecule on the T cell binds to the MHC class II, stabilizing the interaction. This recognition, along with costimulatory signals, activates the helper T cell, initiating adaptive immune responses. This diagram shows how infected cells process endogenous antigens, particularly viral proteins, for presentation to cytotoxic T cells. This pathway operates in all nucleated cells. When a virus infects a cell, it takes over the cellular machinery to produce viral proteins in the cytoplasm. Some of these viral proteins are tagged for degradation by ubiquitin and are broken down by proteasomes into short peptides, typically 8 to 10 amino acids. These peptides are transported into the endoplasmic reticulum through special transporter proteins called TAP, which stands for transporter associated with antigen processing. Inside the ER, newly synthesized MHC class I molecules are waiting. These molecules are stabilized by chaperone proteins, including calnexin and calreticulin, until appropriate peptides bind. When viral peptides enter the ER, they bind to the groove of MHC class I molecules. This binding stabilizes the complex, allowing it to be transported through the Golgi apparatus to the cell surface. On the cell surface, the peptide MHC class I complex is recognized by cytotoxic T cells. The CD8 molecule on these T cells binds to MHC class I, stabilizing the interaction. If the T cell receptor recognizes the viral peptide, the cytotoxic T cell becomes activated and kills the infected cell, preventing viral replication and spread. This table summarizes the key differences between MHC class I and class II molecules. Understanding these differences is crucial for understanding immune responses. Distribution differs significantly. Class I is present on all nucleated cells, enabling immune surveillance of every cell in the body. Class II is restricted to professional antigen-presenting cells like dendritic cells, macrophages, and B cells, thus limiting where exogenous antigens can be presented. Structure differs in the chains involved. Class I consists of a polymorphic alpha chain plus beta-2 microglobulin. Class II consists of both polymorphic alpha and beta chains. The peptide binding groove structure affects what peptides can bind. Class I has a closed groove accommodating peptides of 8 to 10 amino acids. Class II has an open groove accommodating longer peptides of 13 to 25 amino acids. They present different antigen types. Class I presents endogenous antigens from within the cell, primarily viral proteins. Class II presents exogenous antigens from outside the cell, primarily bacterial proteins. Processing pathways differ. Class I uses the cytoplasmic proteasome pathway. Class II uses the endosomal lysosomal pathway. Finally, they activate different T cell types. Class I presents to CD8 positive cytotoxic T cells that kill infected cells. Class II presents to CD4 positive helper T cells that coordinate immune responses. Transplantation is the transfer of living cells, tissues, or organs from one individual to another, or from one site to another in the same individual. Understanding graft classification helps predict transplant outcomes. Autograft involves transplanting tissue from one location to another in the same individual. Examples include skin grafts for burn victims or using a vein from the leg in coronary bypass surgery. Since the tissue is genetically identical, there's no rejection. Isograft involves transplanting between genetically identical individuals, specifically identical twins. Like autografts, isografts are not rejected because MHC molecules are identical. Allograft involves transplanting between genetically different individuals of the same species. This is the most common type of clinical transplant, including kidney, heart, or liver transplants between different humans. These grafts face rejection because of MHC differences. Xenograft involves transplanting between different species, such as pig heart valves in humans, or attempting to transplant pig organs into humans. These face the strongest rejection because of both MHC differences and other antigenic differences between species. Modern medicine has achieved remarkable success in transplanting various organs and tissues. Let's review the commonly transplanted organs. Kidney is the most frequently transplanted solid organ, treating end-stage renal disease. Transplanted kidneys can function for decades with proper immunosuppression. Cornea transplants restore vision in patients with corneal damage or disease. These have high success rates partly because the cornea is relatively immune-privileged with limited blood supply. Liver transplants treat liver failure from various causes, including cirrhosis and hepatitis. The liver has some unique immunological properties that sometimes allow reduced immunosuppression. Bone marrow or hematopoietic stem cell transplantation treats blood cancers, immune deficiencies, and certain genetic diseases. This unique transplant can cause graft versus host disease, since the transplanted immune cells can attack the recipient. Heart transplants treat end-stage heart failure. Recipients require lifelong immunosuppression. Skin grafts treat severe burns and wounds. Lung transplants treat end-stage lung diseases like cystic fibrosis or COPD. Pancreas transplants, sometimes including islet cell transplantation, treat type 1 diabetes. Each organ presents unique immunological and surgical challenges, but all require careful MHC matching when possible, and immunosuppression to prevent rejection. Graft rejection can occur at different time points after transplantation, each with distinct mechanisms. Hyperacute rejection occurs within minutes to hours of transplantation. Acute rejection occurs between 7 and 21 days after transplant, though it can occur months later if immunosuppression is inadequate. Chronic rejection develops over months to years. It's characterized by slow, progressive organ failure from both cellular and antibody-mediated mechanisms. Graft versus host rejection is unique to transplants containing immunocompetent cells, primarily bone marrow transplants. Let's examine a case illustrating hyperacute rejection. John, a 45-year-old dialysis patient with a history of blood transfusions, undergoes kidney transplant that initially appears successful during surgery. However, within hours, the transplanted kidney shows alarming signs of failure. The kidney exhibits swelling and discoloration, appearing dark instead of healthy pink. There's no urine production, despite adequate hydration. His blood pressure rapidly decreases, indicating cardiovascular compromise. He experiences severe pain at the transplant site and progresses to organ failure. This catastrophic response occurs because John has preformed antibodies against the donor kidney's antigens, likely from his previous blood transfusions. The moment blood flow is established to the transplanted kidney, these antibodies immediately bind to the graft's blood vessels. The antibodies activate the complement system, causing immediate endothelial damage and thrombosis in the graft's blood vessels. Within hours, the kidney becomes non-functional due to loss of blood supply. This case illustrates why thorough pre-transplant screening is critical, including cross-matching tests to detect preformed antibodies. If antibodies are detected, that particular donor cannot be used. Hyperacute rejection cannot be reversed once it occurs. Let's understand the immunological mechanism of hyperacute rejection in detail. Hyperacute rejection occurs within minutes to a few hours of transplantation. The hallmark is the presence of preformed antibodies in the recipient's blood directed against donor antigens, particularly MHC molecules, or ABO blood group antigens on the graft endothelium. These antibodies are most likely generated from previous exposure to foreign antigens through prior transplants, blood transfusions, or multiple pregnancies. The immune system has already been sensitized and has memory B cells producing these antibodies. When blood flow is established to the transplanted organ, these circulating antibodies immediately bind to antigens on the graft's blood vessel walls. This antibody binding activates the complement cascade, generating complement components including C3a and C5a. This is why cross-matching is mandatory before transplantation. Let's examine acute rejection through Jacob's case. 18 days after a successful kidney transplant for polycystic kidney disease, Jacob develops concerning symptoms. He notices decreased urine output, indicating declining kidney function. He develops low-grade fever, typically 38 to 38.5 degrees Celsius, suggesting an immune-mediated inflammatory process. He experiences pain at the transplant site from swelling and inflammation of the graft. He feels increasingly fatigued as his kidney function deteriorates. Concerned, Jacob returns to the transplant center, where evaluations reveal several alarming findings. His blood pressure is elevated, often occurring when transplanted kidneys aren't functioning properly. The graft is tender to touch, indicating inflammation. His creatinine levels are rising, indicating decreased kidney function. His urine shows proteinuria, meaning protein is leaking through damaged kidney filters, and white blood cells, indicating inflammation. These findings indicate acute rejection, a common complication occurring in 10 to 30% of kidney transplant recipients despite immunosuppression. The timing between 7 and 21 days post-transplant is classic. Let's understand the immunological mechanism in acute rejection. This is a cellular immune response rather than humoral. The mechanism involves several steps. Recipient T cells recognize donor MHC molecules on the graft cells as foreign. This recognition can occur directly, where recipient T cells recognize intact donor MHC molecules on donor antigen-presenting cells, or indirectly, where recipient APCs present donor antigens to recipient T cells. This recognition leads to direct cytotoxicity. Cytotoxic T cells become activated and directly attack graft cells, displaying foreign MHC molecules. Much of the damage to graft cells results from hypoxia due to interrupted blood flow. The inflammatory infiltrate compresses blood vessels, and endothelial damage further compromises perfusion. Unlike hyperacute rejection, acute rejection can often be reversed with corticosteroids, increased immunosuppression, or anti-T cell antibodies if detected early. Mahima's case illustrates chronic rejection developing over years. She received a kidney transplant for lupus nephritis when she joined medical school. Initially, the transplant functioned well with proper immunosuppression. However, in year 4, subtle signs emerge. Her creatinine slowly rises, indicating declining kidney function. She develops proteinuria as the kidney's filtering capacity deteriorates. Her blood pressure becomes increasingly difficult to control. By her final year, symptoms become more prominent. She experiences fatigue from accumulating metabolic waste products. Her kidney function continues worsening, despite optimized immunosuppression. Her blood pressure remains uncontrolled, despite multiple medications. Ultrasound reveals characteristic chronic rejection findings. Chronic rejection develops over months to years, representing the leading cause of late graft loss. It takes more than 3 weeks, months, or even years after transplanted tissue has resumed normal function. The etiology is likely multifactorial. It may result from accumulated effects of many minor acute rejection episodes that were subclinical or inadequately treated. Additionally, it involves induction of anti-graft antibodies that cause gradual vascular damage. Chronic rejection is characterized by slow and progressive organ failure. In kidneys, specific pathological changes occur. Most patients with chronic rejection eventually require return to dialysis and consideration for retransplantation. Graft versus host disease is a unique, life-threatening complication that differs fundamentally from other rejection types. Let's understand this reverse rejection phenomenon. In typical rejection, the recipient's immune system attacks the donor graft. In GVHD, the opposite occurs. Donor T cells within the graft recognize recipient tissues as foreign and mount an immune attack against the recipient. For GVHD to occur, 3 conditions must be met. First, the graft must contain immunologically competent cells, specifically mature T lymphocytes capable of recognizing antigens and mounting immune responses. Second, the host must possess important transplantation alloantigens, particularly minor histocompatibility antigens, that are lacking in the donor graft. Even when major HLA antigens are matched, minor histocompatibility differences can trigger GVHD. Third, the host must be immunologically incompetent or severely immunosuppressed, incapable of mounting an effective immune response against the foreign donor T cells in the graft. This prevents the recipient from rejecting the attacking donor cells. The classic example is bone marrow transplantation, where the graft inherently contains many immunocompetent T cells that become engrafted and can attack recipient tissues. Remarkably, GVHD can occur even when HLA antigens are perfectly matched between donor and recipient. This occurs because of minor histocompatibility antigens. These are proteins encoded by genes outside the MHC complex that differ between individuals. The mechanism involves donor lymphocytes recognizing recipient minor histocompatibility antigens as foreign. These activated donor T cells recruit recipient inflammatory cells, including macrophages and neutrophils. Ironically, the recipient's own inflammatory cells become the chief mediators responsible for tissue destruction, recruited and directed by the attacking donor T cells. The degree of HLA matching significantly impacts transplant outcomes. Understanding which HLA antigens are most important guides donor selection. The 3 Class I genes, HLA-A, HLA-B, and HLA-C. They serve as main targets of graft rejection. However, they're not equally important. HLA-B is the strongest transplantation antigen. Mismatches at HLA-B loci most significantly increase rejection risk and decrease graft survival. This makes HLA-B matching most critical. HLA-A is much stronger than HLA-C in triggering rejection, but weaker than HLA-B. HLA-A mismatches do affect outcomes, but less severely than HLA-B mismatches. HLA-C is the weakest of the 3. Its impact on graft survival is minimal compared to HLA-A and HLA-B. For this reason, HLA-C is often not tested for matching in routine transplant protocols, focusing resources on the more important HLA-A and HLA-B. A mismatch in these genes, particularly HLA-B and HLA-A, stimulates production of cytokines and enhances immune responses against the graft. The more mismatches, the stronger the rejection response and the more immunosuppression required.